My name is Maren Friesen. I'm a associate professor in the Department of Plant Pathology at WSU. I've been here for 7.5 years now. And today I'm going to be talking about two of my favorite things, nitrogen and microbes. So, by way of introduction, the air that we're all breathing right now is comprised of 78% nitrogen gas. However, because of the nitrogen-nitrogen triple bond, this nitrogen that is very abundant is actually not accessible to plants and, by extension, animals. So there's only three known processes that can convert nitrogen gas into biologically available nitrogen. The first is lightning, which contributes about 5 teragrams of nitrogen per year into the global nitrogen cycle. Biological nitrogen fixation is carried out by this really super cool enzyme called nitrogenase that has some different forms and yeah, super interesting, fascinating. It's currently only found in bacteria and archaea. And in natural ecosystems, biological nitrogen fixation contributes another 58 teragrams of nitrogen. And then in agricultural systems, we get about that much again. So an additional roughly 60 teragrams of nitrogen per year. And then far and away, the largest contributing amount of nitrogen is through the Haber-Bosch process, which generates synthetic nitrogen fertilizer, which again doubles the total amount of nitrogen going into our nitrogen cycle. And it's not an understatement, I would say, to claim that synthetic nitrogen fixation really changed our world. So I like showing this graph just for context. So about half of the global population basically owes their lives to the invention of synthetic nitrogen fixation in terms of food supply that has been enabled through the invention and then widespread adoption and development of crop varieties that can use this synthetic nitrogen during the Green Revolution. However, as we are recognizing more and more, there are a number of potential problems with widespread nitrogen fertilization. So the first for farmers, obviously, is cost. Nitrogen fertilizer can be expensive. The prices can fluctuate wildly because they're tied to other global markets. And then particularly in the inland PNW, there's significant issues with nitrogen fertilization causing a reduction in soil pH. And so these issues sort of motivate the question of whether or not we can replace some or maybe one day all of synthetic nitrogen fertilizer with biological nitrogen fixation. So when we think about the nitrogen pools in this global nitrogen cycle, we can kind of simplify it down into this big atmospheric pool that then gets fixed into ammonia, NH4, that then goes into nitrate, and then all of this goes into organic matter. So all of these processes are driven by microbes, primarily in the soil. So nitrogen fixation, nitrification, and denitrification. And under nitrogen fertilization, there are pretty dramatic shifts in both the rates of these processes, as well as in some cases of the population responses of the microbes. So when synthetic nitrogen is added to a system, nitrogen fixing bacteria will downregulate the amount that they're fixing. So those rates will typically drop. The population responses don't seem to change immediately. This is a meta-analysis of data that some colleagues and I put together a few years ago. Adding synthetic nitrogen to a system dramatically increases rates of nitrification as well as denitrification, and also increases the prevalence of the microbial functional genes that carry out these processes, particularly ammonia oxidizing bacteria, which increase over 300%, which is pretty wild. So my lab's research program is to understand the ecology, evolution, and genomics of plant-associated nitrogen-fixing bacteria and communities in order to harness these interactions for the benefit of humanity and our planet. And I love this cartoon of a bacteria as the Thor of soil, cleaving the nitrogen-nitrogen triple bond with its nitrogenase hammer and using the energy of ATP. So when we think about trying to manage nitrogen fixation, so in plant pathology, there's this disease triangle. So we can apply the same sort of logic to beneficial functions like nitrogen fixation, where we have our microbe, the diesotroph, the host plant, and then the environment. And the environment can include climate, soil management, cropping systems, other members of the microbiome. And we can think about nitrogen fixers broadly in these two classes. Associative nitrogen fixers versus symbiotic nitrogen fixers. And my lab has worked in both of these systems. So associative nitrogen fixers are typically free-living bacteria that live in association with the rhizosphere, sometimes in above-ground tissues or endophytically as well, but many of them will live in the root zone, so they're proximal to the root, and they fuel nitrogen fixation through carbon sources that are secreted by the plant roots. Symbiotic nitrogen fixation is also typically, there's some above ground, but many of these symbioses occur below ground with roots. And so in these systems, the plants will produce these special root organs called nodules, which house specialized bacteria that actually basically turn into nitrogen-fixing organelles within the plant cell. And so they can fix very, very high rates of nitrogen in symbiosis, much higher than the rates that have been measured for associative nitrogen fixation. But both of these processes can contribute substantially to nitrogen in our agroecosystems. So a project that I was part of when my lab was at Michigan State University, we measured free-living nitrogen fixation rates in association with switchgrass rhizospheres in marginal lands. And we found that the primary environmental driver of these rates was soil moisture. So after rainfall events, nitrogen fixation rates went up. So showing, and there's obviously lots of scatter in this graph. But just, showing that things like rainfall can play really key roles in these rates. Other work in my lab has worked on intercropping. So I mentioned that symbiotic nitrogen fixation can fix really high rates. In intercropping, some of this nitrogen can potentially be used by non-leguminous crops. So in this case, canola has higher levels of tissue nitrogen when it's grown intercropped with pea than when it's grown in monoculture. So I have come up with a set of potential practices to promote nitrogen fixation, just thinking through, sort of my knowledge of the biology of nitrogen fixing systems and, my knowledge of things that folks in this room know a lot more than I do about in terms of farming. So the first is potentially fertilizing with particular metal cofactors that are required for these enzymes and that are sometimes limiting in soil. So these could be iron, molybdenum, less commonly vanadium. Reducing tillage can promote free-living nitrogen fixation by creating these little microhabitats that are beneficial for nitrogen-fixing organisms to live and fix nitrogen within. Adding organic nitrogen can potentially prime systems for nitrogen fixation. Planting legumes can contribute, these specialized root nodule habitats for symbiotic nitrogen fixation. Compost tea could, in principle, enhance the population or potentially the diversity of nitrogen-fixing communities. And then there's always inoculants. We could just add directly the nitrogen fixers that we want in a particular system. So to explore in more detail these connections between the environment and management, nitrogen fixing diversity, and actual nitrogen fixation rates and processes, I have a number of active projects in my lab. So some of these are funded by the USDA, some by the Oilseed Commission and the Grain Commission. So I'll just give you a very, very brief overview. These are active, ongoing projects. So we are sampling soils, and this is in collaboration with the LTAR Cook Farm. So with USDA collaborators, we have a little bit of data that I will show in a moment, where we're comparing the long-term aspirational to the business as usual soils in terms of the diesotroph diversity. We're also comparing agricultural and nearby prairie sites around the Pullman area. And then we're collaborating with the Washington Soil Health Initiative baseline survey, as well as the Soil Health Institute North America-wide soil health surveys to characterize the diazotroph communities within these different scales. So what do we know so far? So we're actively extracting and sequencing tons of samples, and I think my postdoc Javi is here. So he's been working diligently on this. So we know that we are able to see quite diverse diazotroph genera. So this is from Cook Farm, comparing the no-till and the business as usual sides. So there's many diazotrophs present in these systems. The identities of the dominant taxa on the no-till versus the business as usual side are different. When we look at these communities, the communities are very clearly different when we ordinate them. And when we look at the overall diversity, the business as usual side is actually more diverse. So there's more potential nitrogen fixers on that side. And then we've started pulling out these correlations between the genera that we detect in terms of the diazotroph communities and soil health measurements that Tim Politz and other USDA colleagues have measured for these samples. So my long-term vision and my motivation to speak to you guys this morning is that, really I would like to use some of my lab's knowledge and expertise to create a hub for innovative and integrative research, training, and engagement centered around really trying to leverage and use microbiomes to benefit plant, soil, and societal health. And so one way that I would like to try to do this is by putting together a sustainable agricultural systems proposal. So this is in very early development, so we're seeking feedback and partners to basically try to ground truth some of these biofertilizers that are on the market today. So it's a very large and rapidly growing market. About 79% of the products available today are nitrogen fixers. Another 14% are phosphate solubilizers. And this is a picture from up outside Spokane of a couple of farmers who let me take some samples of their compost extract that they were putting on their fields. And just an example of a biological inoculant product that some people in our area are using. But so people are trying these things, and we don't really understand how applying these products are shifting the ecology of the system, the cases in which they work or don't work. And there are potential downsides of microbial inoculants. So a few years ago, some of my postdocs and other collaborators wrote an opinion paper about how widespread use of inoculants that we don't really understand could potentially have unintended consequences on, sort of the rest of the agroecosystems that we're putting them in. And, really sort of proposing that we both understand the microbes that we're adding to our systems, as well as monitoring them to understand how they work and why. So for this potential sustainable ag systems project, I've been chatting with Mike Strickland at the University of Idaho, who couldn't make it this morning. But we are considering trying to put together some inland PNW-wide trials of nitrogen fixers, as well as potentially biological nitrification inhibitors. Monitoring microbiome and soil health, and then working really closely with farmers in terms of co-innovating the management of these microbial products, and trying to understand what we need to know in terms of the basic science, but then also in terms of like what data and evidence farmers need to be able to make the decisions to either use these products or not, and in ways that would be useful for application. So in terms of a overall outlook from this brief presentation, in terms of reducing synthetic nitrogen fertilizer with microbes, I think that there are many potential diverse approaches and not one silver bullet. Overall, I'm very much in favor of trying to manage the ecology of our agricultural ecosystems to promote the growth and functioning of existing and or inoculated nitrogen fixers. I think it's really important for us to work towards understanding when and how inoculants and compost teas and extracts actually work. And this will require, I think, a lot of farmer and researcher co-innovation. And for discussion, since we're going to have discussion for the rest of this hour, I'd really like, feedback from all of you in terms of, what do you think is needed in terms of, moving towards replacing synthetic nitrogen with biological nitrogen fixation, either on your operation or in our area. So with that, I'd like to acknowledge my sources of funding, collaborators, and all the great folks in my lab, past and present. So